So what we aim to do is now go for a more complex system where we put the cells together in the same system, so blood-brain barrier with microglial cells, and see how they crosstalk each other. I'm Daniela from Nova Institute for Medical System Biology. I'm a PhD student and I'm focusing on understanding how gut polyphenols metabolites influence neuroinflammation and brain health. And more specifically, we are using a microfluidic system to study how these compounds interact with the BBB microglia crosstalk. We know that neurodegenerative diseases are being increasing prevalent, and we know that neuroinflammation it's one of the main hallmarks common to most of the neurodegenerative disorders. So we have particular interest in this topic, and our compounds, our gut polyphenols metabolites, already show some results that could improve this phenotype of the disease. So in this case, and we also know that our compounds could per se improve BBB features. So what we aim to do is now go for a more complex system where we put the cells together in the same system, so blood-brain barrier with microglial cells, and see how they crosstalk each other. And yeah, this is the main focus of my research. Since microfluidic systems, it's really difficult to implement in our lab. We begin for a simple model. So we actually begin with single cells in different plates, so human microglial cell lines and microglia in different plates, and we saw some interesting results that they could decrease the release of pro-inflammatory markers. And in the case of human endothelial cell lines, we saw that they actually could decrease the number of ZO1 gaps in the membrane of endothelial cell lines. And in the case of microglia, our compounds were able to reduce the nuclear translocation of NAKB. When we put the cells together in a local culture system, we also saw that we don't have a barrier leakage, so that seems that our barrier is intact, and we also could see that the cells are cross-communicating each other. So when we add our compounds in one compartment, we see a reduction of a pro-inflammatory marker in the other compartment. So this is the main results that we have now. We just begin now implementing we just begin now having the first results with the microfluidic system. So we have this system in the lab. We use the chips from Dynamic 42 with two membranes, where we seed the brain endothelial cell lines in the upper compartment and microglial cell lines in the lower one. And after 1 day, we begin our experiment with a pump, and we incubate with our compounds and give a pro-inflammatory mixture. And yes, so this is the first results that we got with the chip, showing that actually our model is working. So we have a difference between control versus the pro-inflammatory mixture. So we have, and we also have difference between ZO1. So we saw that the membrane of the cells in the mixture, it's more, so we have more gaps when we control to the control, that we have a more tight membrane with a more reorganized membrane. Yeah, so this is the preliminary results that we got in our system. What we aim to do with this system, so we know that our compounds reach the circulation, not just one, but more than one compound at the same time. In this time, we are using just single compounds, static conditions. So what we aim to do in the future is to combine these polyphenols and do like a pharmacokinetic profile in the chip. I think the first thing that you have to do is really know what the research question of your work. So what you aim to do, the outputs that you need. I would say that this kind of World Summits of MPS, where we could meet other people that already work in the field and see the booths, feeling the chips, asking questions. I think it's the tips that I could add.